and so on. Okay. So the second topic will be peptide hormone. Okay. If you look back at the topics of the peptide hormone, okay. So all the topics relating to the peptide hormone will be included in test one. Okay. So you have to look at the types and function, bias disease, okay, receptor and pathophysiological defect. Okay, so the number 4 pathophysiological defect is about the disease. Okay, you have to look into the disease that relate to the peptide hormone. Okay, and for the last one, the last topic for the test one will be glycoprotein hormone. Okay, I think this one is we just talked about it last week. Okay, I think must be still fresh in your mind. Okay, so that one will be included in the test one. Okay, so you have to know the type and function, reaction mechanism, and so on. Okay, so far, do you have any questions regarding the topics that will be included in test one? All right. So if you don't have any, yes.
What about the format of the test?
Okay, relax, relax. Okay, we go one by one. Okay, so I will explain about the topic first. Okay. Okay, so if you don't have any questions about the topics, okay, so now we move on with the format. Right. So this is the format for the test one. Okay. So for test one, we have two sections. Okay. So we have section A and section B. Okay. So for the section A, design for the objective questions means you have to choose just A or B or C or D. Okay. So you just pick one, the correct answer. Okay. So usually for the objective question for section A, okay, I plan to have 40 questions, but it's not finalized yet. Okay. I'm not sure whether I managed to get 40 questions or just 30 questions. So the numbers of the objective question will be around 30 to 40 questions. Okay. So the marks also 30 to 40 marks, depending on the how many questions for the objective questions. Okay, for the section A. Okay. So for the section B, okay, mostly the question B is about the essay questions, but I think one or two questions it's just a short essay questions. Okay, so there are 4 questions in section B. Okay, so the mark will be around 30 marks. Okay, for 2 hours you have to answer section A and section B. Okay, so the total mark will be around 60 to 70 marks. Okay, so we will make a percentage how many questions, how many marks you get for this test one, and we'll make it as 100%. Okay, so out of 70, how many marks you can get? Okay, so far, how do you have any questions regarding test one?
Doktor, section B tu kena jawab semua ke? Ke ada
Yes.
pilihan?
No. For the test one, for the section B, we don't have any choice. Okay, so you have to answer all the questions. Okay, so but starting from the test two, we'll give the choice for the section B. Okay, because the test one is quite straightforward actually. Okay, so I think most of you will score more on section B compared to section A. Okay, because section B is just straightforward. If you read the the lecture note, you can answer the section B. Okay, what you have to do just read the the lecture note for the section B. Okay. So any other questions?
Doctor, for the short essay,
Mhm.
it's like roughly like what, like 5 marks or
No.
for each?
For the for the essay, the long one maybe around 10 to 12 marks. Okay, that maybe for the long one, maybe I want you to explain about the mechanism. Okay, for example, so will be a lot of mark from there. Okay, so maybe 10 to 12 marks. But for the short essay, usually just I just need you just a single paragraph for the answer. Maybe just 2 marks. Okay, that one is just a short essay. It's not short essay actually. I'm not sure what to call it. It's just I give you question, you just answer it, just like a suggestive question, something like that. Okay, it's not even an essay. It's actually for the short one. Any other questions? Okay, all right. So if you don't have any questions, okay, maybe we can start back to our come back to our lecture. Okay, so I will share the slides. All right. Where's the slide? Okay. So this is slides. Okay. So for today, we continue with the biosynthesis of the, I believe, sorry, of the thyroid hormone. Okay. Okay. So okay, for the I forgot to tell you for the format and the topics that included will be included in test one. Okay. So the slide I will upload in the Putra Blast. Okay. So if you want to refer back the format and also the topics, you can just go to the Putra Blast. I put the slide about it on the Putra Blast. Okay. So you don't have to worry. Okay. Besides that, if you still have any questions, you just contact me through WhatsApp. Okay, asking about the text one. Okay, so okay, now we continue with the topics. Okay, right. So for today, I just have one topic to talk about, which is the biosynthesis of thyroid hormone. Okay, so after we finish this topic, the biosynthesis of thyroid hormone, I think we can continue the class for next week. Okay, because I got emergency thing to do. Okay, I have to pick up my car at the workshop after this. Okay, so maybe I just cover these topics for today. Okay, so for the biosynthesis of thyroid hormone, like I said on Monday, thyroid hormone actually derived from single amino acid. Okay, which is the tyrosine. Okay, so from the tyrosine, if you modify the tyrosine, you can get the thyroid hormone. So today what we want to talk about is about how this tyrosine can be converted into the thyroid hormone. Okay, so the first mechanism in order to convert the tyrosine into thyroid hormone is the iodine trapping. Okay, as you know, okay, the difference between tyrosine, the amino acid of tyrosine, and thyroid hormone is the addition of iodine. Okay, so if you add iodine into the tyrosine amino acids, then this tyrosine actually can be converted into thyroid hormone. They have the activity. Okay, so the ion trapping made possible by iodide pump. So how these cells, the cell in the thyroid gland, want to get the iodine? So iodine actually comes from your diet. Okay, if you eat something that contains iodine, like seafood, okay, so the iodine will be released in your blood circulation. In order to get this iodine in the blood circulation, okay, the thyroid gland, the cell in thyroid gland, actually they have a pump, which is iodide pump. The function of this pump is to get or to pump in the iodine from the blood into the cells to be used as the precursor for the production of thyroid hormone. Okay, so the name of the pump is sodium iodide synthetase. Okay, and also this pump actually need energy. So you know, right, I think I'm not sure if, if you have taken biochemistry of protein. Okay, but in biochemistry of protein, okay, we have talked about the what call it, different pump system in our body. Okay, so actually the pump system in our body can be divided into two. Okay, the first one is the one that can be used without any energy, whilst the second one is you have to use energy in order to make the pump to be active. Okay, so we have active and inactive transporter. Okay, so for iodine, in order to get the iodine from the blood, we need the pump that need energy in order to work. Okay, so the name of the pump is sodium iodide synthetase. Okay, so the energy comes from the, we call it the ATP. Okay, then the second part, in order to make a thyroid hormone, is thyroglobulin synthesis. Okay, if you imagine back, okay, the cells in our thyroid glands, okay, at the middle of our cells, we have a colloid, okay, like a space in the middle of the cells. Okay, inside the colloid, actually we have thyroglobulin. Okay, actually this thyroglobulin will be used to produce thyroid hormones. Okay, so thyroglobulin is a protein that contains a lot of tyrosine. Okay, so actually the source of tyrosine or the thyroid hormone production comes from the thyroglobulin. Okay, so thyroglobulin is a precursor for T3 and T4. So you know that we have three different types of thyroid hormone: T3, T4, and also reverse T3. Okay, so all of them actually come from the thyroglobulin. Thyroglobulin, the one that provide the tyrosine. Thyroglobulin is produced in the ribosomes like other proteins. Okay, so you know that in the protein synthesis, the mRNA comes from the nucleus, then will attach to the ribosome to translate the protein. Okay, so similar thing with thyroglobulin, since thyroglobulin is a protein. Okay, so the thyroglobulin actually produced by ribosome inside the cell in the thyroid gland. Okay, so the cell produces thyroglobulin and export the thyroglobulin into the colloid, the space in the middle of the cell. Okay, so the colloid actually is the like storage of this thyroglobulin. Okay, then thyroglobulin is released into the follicular lumen. Okay, so follicular lumen actually is the colloid. Okay, so after the cells, okay, producing the thyroglobulin, then this thyroglobulin will be exported into the lumen, the colloid lumen. Okay, or we call it as the follicular lumen. Right. Then what happen is, so now inside our cells, we have thyroglobulin. We have source of tyrosine. Okay. What we need is the iodine. So the iodine will be taken from the blood. Okay. So iodine movement diffusion throughout the follicle cell. So in order to get this iodine, the iodine must go inside the cell. Okay. From the blood circulation, the cell must take up this iodine inside the cells. Okay. So then after they take this iodine inside the cells, it will exit from the apical membrane by iodide transporter called pendrin. Okay, so after the cells, okay, absorb the iodine using the pump, the iodide pump, okay, inside the cells, then what happens to this iodine? It will transported again into the colloid or follicular lumen. Okay, so because the process of the synthesis happen in the colloid, not in the inside the cells. The cells just provide the thyroglobulin and exported into the colloid. Then the iodine from the blood circulation will go inside the cell, then transported also into the colloid or the lumen, follicular lumen. Then when we have all these things, okay, inside the colloid, then the conversion of the tyrosine into the thyroid hormone can happen. Okay, the process we call it as organification. Okay, which first the iodine have to be oxidized, okay, by enzyme we call it thyroid oxidase. Okay, so the iodine need to be oxidized. Oxidized means this iodine need to get some oxygen. So where the oxygen come from? The oxygen come from the peroxide. Okay, so actually peroxide is a free radical. Okay, free radical is something that very active to attack any other compound. Okay, so the free radical comes from the metabolism in our body. So most of the time, okay, when our body want to generate ATP or energy, the process happen in the mitochondria. So the process generating energy actually producing free radical. This free radical can be used by the thyroid cells to produce thyroid hormones. Okay. Then after that, after the iodine being oxidized, what happen is Thyroglobulin will be linked with this iodine, okay, by thyroperoxidase or TPO. Okay, so we need two process. First, you have to oxidize the iodine. The second one, after the iodine being oxidized inside the lumen or colloid, then this iodine have to be attached to the thyroglobulin. Okay, this attachment produce monoiodotyrosine or MIT or diiodotyrosine. Okay, so depending on how many iodine attached to the thyroglobulin, okay, it can produce either monoiodotyrosine, means inside the thyroglobulin we have a lot of tyrosine. Then depending how many iodine bind to the tyrosine, it will be classified into two, either MIT or DIT. Okay, if just one iodine attached to the tyrosine inside the thyroglobulin, then we call it as the MIT or monoiodotyrosine. But if there is two iodine, okay, bind to the tyrosine inside the thyroglobulin, we call it as diiodotyrosine. Okay, then we have another process which is the last one, coupling. Okay, that they may also become T3 and T4 soon by TPO. So after we have the MIT and DIT inside the thyroglobulin, then this MIT and DIT will couple each other, okay, will bind each other to form thyroid hormones. Okay, so if you want to produce T3 or hormone, thyroid hormones, then MIT will bind with DIT. Means we have three iodine in total. MIT contains one iodine while DIT contains two iodine. When they bind or couple together, they can form T3 hormone. Okay, T3 hormone, different between T3 hormone and T4 hormone is the numbers of the iodine. T3 hormone, they have 3 iodine, while T4 hormone, they have 4 iodine. Okay, so the reaction of coupling also catalyzed by the enzyme, we call it thyroperoxidase or TPO. Okay, so this is the what call it the summarize about the whole process. Okay, so we start from the first one. Okay, I think by explaining the figures, more understandable compared to reading the words, right? Okay, so try to explain this one. Okay, so the first mechanism is the absorption or the intake of iodine. Okay, from here at the bottom, if you can see NIS, there is NIS. Okay, NIS stand for sodium iodide synthetase. That's the pump that pump the iodine from the blood inside the cells in the thyroid gland. Okay, so this pump, in order to work, it needs two compounds to bind together into the pump before you can activate the pump. Okay, the compound is iodine itself and also the sodium. Okay, if there is no sodium, okay, the iodine cannot enter the cells because the pump needs sodium and iodine in order to work. Okay, then what happens is the iodine will come inside the cell. Okay, then inside the cells, this iodine will pump out into the colloid or follicular lumen, the space inside the thyroid cells. Okay, there's space inside in the middle of thyroid gland, the thyroid cells. Okay, if I can show you, okay, try to show it. Okay, so this is the colloid. Okay, so the colloid at the middle of this, okay, actually the place where the thyroid hormone being synthesized. So the iodine from outside will pass through the cells and go to the colloid, okay, or the follicular lumen. Okay, and okay, then the iodine, in order to go to the colloid or follicular lumen, you need another channel, okay, in order to pass through the membrane of the cells. Okay, the channel we call it spandrene. Okay, you can see the P here. Okay, the orange one actually is the channel for the iodine to go to the colloid or the follicular lumen. Okay, then what happens is we have also thyroglobulin that being produced in ribosome. Okay, then will be modified in Golgi apparatus and before it engulfed by the vesicles. Okay, then this vesicle will bring this thyroglobulin also inside the colloid or lumen. So now inside the lumen, we have two main components for the production of thyroid hormone, which is the thyroglobulin and also the iodine. Okay, and also the iodine, once they go to the lumen or colloid, okay, this iodine also being oxidized by TO or thyroid oxidase. Okay, another enzyme that will oxidize this iodine. Okay, so the oxidation occurs because the availability of the peroxide. H2O2 actually is the peroxide. Okay, right. Then this peroxide will donate one oxygen into the iodine, okay, to make the iodine reactive. Okay, once the iodine receive oxygen, okay, then it will become reactive. Okay, so means reactive, it can easily bind to any other molecules. Okay, for this case, the iodine, once they receive the oxygen, it become reactive. It can easily attach to the thyroglobulin. Okay, then now we have reactive iodine and we have also thyroglobulin inside the lumen or colloid. Then what happen is another enzyme which is TPO. Okay, thyroperoxidase enzyme. Okay, the function of the enzyme is to attach this iodine, to catalyze the attachment of this iodine with the thyroglobulin. Okay, so depending on the numbers of iodine that attach to the tyrosine of the thyroglobulin, we can produce either DIT or MIT. Okay, DIT means diiodine thyroglobulin. Okay, means it has two iodine in the tyrosine. Okay, well, for MIT, just have one iodine. Okay, right. Then what happen is we have DIT and MIT in the similar in the thyroglobulin. Then this DIT and MIT will be absorbed back inside the cells, okay, by endosome. Okay, this endosome will engulf this DIT and MIT. Okay, then this endosome will go inside the cells. Okay, then inside the cells, this endosome actually contains a protease, a lot of protease actually. This protease, the function is to cut all these thyroglobulin and MIT and DIT. Okay, then after cutting, there will be a coupling. Okay, MIT will be coupled with the DIT or DIT will be coupled with MIT to produce thyroid hormone T3 or T4. Okay, then this thyroid hormone will be released into the blood circulation back, going to the organ that targeted. Okay, so then the remaining of the product after the protease digestion, okay, will be reused back for the next production of thyroid hormone. Okay, so this is the overall mechanism how our cells is producing the thyroid hormone. Okay, so far, do you have any questions so far? Right. So actually, I'm not sure why this Zoom give me a reminder that I just have remaining time about 8 minutes before the Zoom cut off. I'm not sure. So we have another 8 minutes before the Zoom will shut off automatically. Okay. So if you have any questions, please ask. Okay. Yes. Okay. The organification. Okay. Here. Okay. So the organification actually the process where the iodine attached to the thyroglobulin. Okay, so the process up here is the organification, where the iodine will be oxidized. Before you attach iodine to the thyroglobulin, you have to oxidize the iodine. If not, this iodine cannot be attached to the thyroglobulin. So the whole process, attachment of the iodine to the thyroglobulin is the organification. Okay, we call it as the organification. Right? Okay, so here the example how the DIT and MIT can be coupled. DIT and DIT can be coupled to make T4 thyroid hormone. Okay, so for example, above here we have thyroglobulin. Like I said before, thyroglobulin we have a lot of tyrosine amino acid residues inside the thyroglobulin. Okay, so this amino acid residues, okay, the tyrosine 1 will be attached to the iodine through the organification. Okay, so depending how many iodine attached to the tyrosine, okay, it can be MIT or DIT, monoiodide thyroglobulin or diiodide thyroglobulin. Okay, for this example, for this particular example, we have two DIT. Okay, we have two tyrosine that being attached by two iodine. Then what happen is inside the what call it, the endosome. Okay, inside the endosome. Okay, after the all this being engulfed back inside the cells by endosome, inside the endosome will be a lot of protease that will cut, okay, all these things. Before the cutting, okay, this DIT and DIT will be attached together by an enzyme. We call it as TPO. Okay, TPO will attach DIT and DIT. Then after the attachment, the endosome will cut the forming T4 and before releasing it into the blood circulation. Okay, so this is the whole scheme how the our T4 being produced. Okay, so for example, if we have at the first place we have MIT and DIT, the final product will be T3. Okay, so depending on the how many iodine being attached to tyrosine, we can produce either T3 or T4 for thyroid hormone. All right, then the storage and excretion of thyroid hormone at follicular lumens. Okay, follicular lumen is the colloid. Okay, MIT and DIT in the thyroglobulin comprise about 90% of total pool of iodine in our body. Actually, most of our iodine being stored in the thyroglobulin. Okay, so it just need a time before it can be converted into the thyroid hormones. Like I said before, thyroid hormone is very important in our body because it controls everything, the metabolism and energy expenditure and so on. So that's why there needs a pool of all the precursors needed to produce the thyroid hormones. Okay, then secretion. Okay, so we have endocytosis and also exocytosis. Endocytosis is the process where the thyroglobulin from the colloid being engulfed back inside the cells. Okay, that one we call it as endocytosis before the final refinement of this thyroid hormone inside the cells. Okay, so the endocytosis, thyroglobulin that attached to iodine is brought back into the follicular cells. Then long pseudodia reach out the apical surface to scoop the thyroglobulin. Okay, there will be like a hand, okay, that will scoop all this thyroglobulin in the colloid before they can be engulfed back inside the cells. Okay, endocytic vesicle migrate towards the basal membrane plus meet the lysosome. Then there will be a lysosome. Lysosome is a protease, contain a lot of protease that will cut this thyroglobulin into T3 or T4. Okay. Then if needed, T3 and T4 release into the blood circulation. Then the remaining of MIT and DIT that been used to produce the T3 or T4 will be recycled back into the colloid. Okay. So like I said before, this is the overall mechanism. Okay, so we just have 2 minutes. Okay, so I think I want to stop the class. Okay, so before we stop, I want to remind you all to sign the attendance at the Putra Blast. Okay, so I already put the attendance. Okay, in the top section of the inside the Putra Blast. Okay, you just have to click that one. Okay. All right, so that's all for today. Thank you for attending the class. Okay, see you next week.
Thank you, Doctor.
Okay, doktor.
Thank you, Doctor.
Thank you, doctor.
Okay.